Good evening, everyone. Thank you so much for joining us this evening for our first virtual Return and Learn presentation of the spring semester. My name is Justin Durensis, and for those of you who do not know me, I serve as Assistant Director of Alumni and Parent Engagement here at the college. And before we get started, I would just like to review a few things this evening that will ensure our presentation not only goes smoothly, but also stays on track with about the hour and 15 minutes we have allotted here this evening. For those that have entered the meeting room tonight, if you have unmuted yourself over the past few minutes, if I can just ask that you go back and click on the little mute icon on the bottom left-hand screen of your window, that would be very helpful. This will help cut down on any background noise. Tonight's event will be recorded, so if you would like to keep your camera on, you may. However, if you would like to turn your camera off, that would be fine as well. This might actually be helpful to limit any excessive movement in the background if you do have your camera off. After the presentation, we will be opening up the session for a live Q&A. So this will be done via the chat box on the bottom of your taskbar. And so if anyone has any questions, I just ask that you submit your question via that chat box to me directly and just let me know that you have one. I will then call on you to ask your question live to Dr. Mosovsky. I would now like to introduce our guest presenter this evening, Dr. Kara Mosovsky. She has been a part of the Moravian community for the past 6 years, and she currently serves as an assistant professor of biology. Her research and interest include antibiotic resistance, intracellular infections, and infectious disease. And so without further ado, I'm going to hand the presentation off to her. But first, before I do that, I'm going to submit a poll to the meeting room that Kara has thoughtfully and very helpfully created so that all of us as attendees tonight can, sort of get a better understanding of what she will be presenting on. So I'm going to launch this poll here in a second. And if I could just give everyone 1 or 2 minutes to complete this poll, just so that Kara can have some ideas of what she'll speak to this evening. So take a minute or two and just answer this question on this poll.
So I'll wait another minute while results are still coming in. I can see the poll results showing up. And I wanted, so first of all, hi, I'm Kara Mosovsky. I'm really excited to be here this evening, and I thank you all for coming out to this Return and Learn for alumni. It's fun to see some old names come up, some older, some not so old. And I'm really pleased to be here. So I figured that the first 30 minutes or so, 25 to 30 minutes, I wanted to give some basic background, basic biology. I know some of us are non-biology majors or never had much biology. And so I'm going to address the COVID and SARS-CoV-2 biology in more simplistic terms. And set the stage so that we can have a really good Q&A afterwards, right? So we'll touch on a bunch of different things. I hope to touch on many of these things in the poll, but I wanted the poll to sort of guide how in-depth I go in this information. So it looks like we've got a lot on the biology of an mRNA vaccine. Obviously, the Pfizer and Moderna vaccines are out and being administered now. So yeah, we'll definitely talk a lot about that, as well as the safety. And there's a lot we don't know. That's great. We can definitely talk about that, and somewhat of a mix of the others here. So I have intended this to be a kind of interactive lecture. Most of my lectures aren't just slides and talking at you. So the way I want to do this is actually to share my iPad screen here. And I have the iPencil, so I'll be able to draw on here as we go along, and you will see my little doodles. But it's going to be a little bit of like a chalk talk, but we have no chalkboard or whiteboard. So again, I'm going to try and set the stage so we can have a good question and answer. And please, as I'm going through, if there's anything that you would like me to explain better, just shoot Justin a quick chat and he'll draw my attention to it. Justin, feel free to interrupt me. And otherwise, write down your questions for in a little bit. All right, so let us begin. So as of 1:22 p.m. today, we had 91 million cases of COVID-19. And our global deaths so far is this number over on here, close to 2 million deaths so far. Right? In the US, we've got 22 million of those cases, and we're about double the number of cases as the next highest country. So we all know of the toll of the pandemic. These are just some numbers. These are our current numbers right now. So I want to start by saying that microbiology is my background and immunology. I do not study the COVID disease in a clinical setting, nor do I study SARS-CoV-2, the virus that causes the disease. Mostly in my research lab, I study bacteria, which is a whole different category of pathogen. But I know a lot about virology. I know a lot about immunity to viruses, and I've read extensively about these topics for the last 10 months. So a little bit about what we know, basic virology here. So a virus, first and foremost, is, let's see if this is, there we go, not a cell, right? So our basic building blocks of humans and animals and plants are cells. Bacteria are single cells. Viruses are not a cell, right? So when you remember cells from, your high school biology, they had a cell membrane, they had a nucleus, right? They had some cytoplasm, they had mitochondria and endoplasmic reticulums. All of that is not present in a virus. So what is a virus? Basically, it is a protein shell, just protein, and inside is some sort of genetic material. This is the heritable piece for a virus. And we have two types of that heritable piece, that nucleic acid. It can either be DNA or RNA. Our coronavirus is, of course, an RNA virus. So this is our coronavirus. We call it, because it's not a cell, we either call it a viral particle or a virion. So this is the basic unit. This is one virus. Here we go. What it is a capsule here of some kind of little fatty material. It's not a membrane, but it's a little bit of a fatty layer. It's got these protein spikes embedded in that fatty layer. And then this curlicue thing on the inside is the genetic material, the RNA, right? So that's all it is. What it can and cannot do, because it is not a cell, it can't replicate on its own. So this virus on a tabletop is just going to sit there. It can't make more of itself on a tabletop, on a surface, in floating through the air. It has none of the machinery to do that, right? So they are obligate parasites. They have to get inside of a host cell before they can replicate, right? So that's a really important thing to remember, is on their own in the environment, they're just sitting there. They can't move on their own. They have no locomotion. They can't jump, right? They can stick to something. That's about it until they get in, and then they use the host machinery and kind of pirate it, take it over to do the replication and make more viruses. All right, so how do they infect us? Again, they can't move. So really, it's a sticking mechanism. So a virus, these little spikes, this is our coronavirus, right? These little spikes on the outside that are just protein are going to bind to a receptor on our cells. And that receptor, if you've heard, is called the ACE2 receptor. And it stands for angiotensin converting enzyme 2, but that's not important. This is the name of the receptor on our cells that this coronavirus can stick to. Once it sticks, it is going to be able to get inside, right? So the reason why you can snuggle with your dog when you're sick and with the flu, for example, and you're coughing on your dog, the dog's not going to get sick because the dog's cells won't be able to bind to human influenza virus, right? These receptors are not going to be able to attach to that virus, right? So here on our coronavirus, we've got these ACE2 receptors, and the spikes on this virus are attaching to our cells' receptors. And where are these receptors? They're not on every cell in our body, but they're on quite a few. So obviously the lungs and the respiratory tract have these receptors. The kidneys do, the intestines, blood vessels, and maybe even a couple others, right? There's certain white blood cells of our immune system, parts of our immune system have these receptors. So the virus, that all of our SARS-CoV-2 viral particles that we're worried about, they all will be able to bind to any cell or tissue that has ACE2 receptor. So again, once they get in, they're infecting. So what happens when they get in? So this is a simplistic diagram. This isn't for coronavirus, but it is for an RNA virus, and more or less they all have the same cycle. So once it gets in, right, so the first step is that attachment. Then, right here, it is going to be taken up into the cell, facilitated by that receptor binding. And it's like that locking key. It's very, very specific. It can only bind to ACE2 receptors. When it does, it will get inside. So it is going to get inside here. And then the piece of RNA, that genetic material, is going to be released into the cell. This blue part here, oops, erase that. This blue outline here is our cell, right? And this purple bit in here is the nucleus inside of one of our cells. In the case of coronavirus, SARS-CoV-2, let's say that this is a lung cell, right? So you breathe in the viral particle, this guy, it attaches to the ACE2 receptor on one of your lung cells. It gets in, the RNA is released, and in order for a virus to make more virus, it needs to make more RNA, the same little strand of RNA that it came in. It needs to make that to go back out, and it needs to make the protein spikes that were around the edge, right? So it was the RNA inside the little lipid envelope, and then these spikes, right? So it's got, if it comes in and it wants to replicate, it's got to make all three of those components to be able to get out. So we have this RNA released inside. It makes these new components. It puts them together, and then it releases the new viral particles, they kind of bleb out from our cell membrane. And this picture is showing one viral particle going in and one coming out, but that's simplified. For every one virus that goes in, hundreds or thousands can come out of one cell, right? If you're breathing in 10 viral particles and they all happen to touch to a cell, this is how we get that exponential growth of viral load very, very quickly, right? So this is happening at a cellular level. But when you're sick with COVID-19, what's happening, right? So we know that there's a lot of asymptomatic people or asymptomatic cases of COVID-19. In the cases that have mild or moderate symptoms, you're having a normal immune response. Your cells are detecting the invader, and you get an inflammatory response. Your immune system kicks in, right? You get an inflammatory and immune response. And what we really want for immunity, I'm sure people know, are antibodies. Everyone's talking about them. We can talk more about where they come from, right? But antibodies are coming from our B cells, which are just one of the types of white blood cells that are part of our immune system. And we know that the antibodies are protective. Again, we can talk more about how they're protective if you all care. But basically, we, in the mild cases, have resolution because our immune system wins, right? And in those people where there is severe COVID symptoms or severity of disease, we find that the inflammatory response is hugely magnified to the point where the immune response itself is actually causing a lot of the damage. It's not the viral replication. Oops, it's not this viral replication cycle so much that is causing the damage. It's actually the immune response that is over-exaggerated and causing harm then to the body. In some studies, they found that actually a really high antibody level in your blood is correlated with more severe disease, which seems counterintuitive, but this is one of the things that they're finding with the COVID cases. All right, so I want to move on a little bit to the biology of vaccination, right? So your immune system is big, and there's really two branches to it. The innate immunity, perhaps this is a review for some of you, but innate immunity is things that you're born with. Right, so certain white blood cells are part of your innate immunity. The mucus that lines your respiratory tract to some extent, right, or your gastrointestinal tract is part of your barrier defenses against pathogens coming in. And these components of innate immunity or your innate protection, there's no memory response, right? So you don't remember if a certain foreign thing comes in, your body isn't able to remember it in terms of this response. And it's very limited to specificity, right? It's not able to determine, oh, that's this strain of foreign virus or bacteria that's coming in. It treats everything foreign as kind of the same, whether it's bacteria or viral, fungal, or protozoan or whatever, right? So it's not specific. Adaptive immunity, this is what we want with a vaccine. And this is what is going to give us protection. If we have COVID, we want this adaptive immunity to stick around for years and keep us protected. So adaptive immunity changes depending on what your body is experiencing in terms of what foreign things am I exposed to. And we have memory here, right? So the B cells, these guys, and the T cells are really important white blood cells as part of our adaptive immunity, right? So the B cell, let's say, I keep touching this by accident. Let's say this is our coronavirus, right? So the coronavirus can activate a B cell, and then the B cell is going to produce antibodies, right? So the antibodies are specific to the virus. So if a different green virus came into our body, B cells, a different B cell would recognize it and create green antibodies. And the green antibodies are not going to do anything to the pink pathogen or vice versa, right? They're color-coded. So this pink antibody is only going to be able to bind to the pink pathogen. And so that is how it's important for us to have an antibody response against the COVID-19 virus, right? And then that is going to be very, very specific to those antibodies are only ever going to be able to bind to the COVID-19 virus. And another part of understanding vaccination biology is a timeline. So let's say this is time and this is antibody level. So going up and time going out. So the first time you are breathing in a COVID virus, let's say that that's here at time 0, you don't make antibodies right away. Antibodies, those B cells, it's going to take them a couple of days to get activated and kick on and start producing antibody. So you won't get antibody produced until usually we say 10 to 14 days after exposure, right? So your antibodies are going to be a little delayed and they're not going to stick around for very long. Those levels are going to drop down eventually. And then at some other point in time, if you get an exposure again to the same exact foreign thing, right, COVID-19 virus, now after this second exposure, your antibody levels go way, 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 way up and very quickly. So there's very little time here. And we get a really, really high antibody level, and it is more sustained, right? So this is what we call a primary exposure to anything foreign. The influenza virus, we could be talking about that. The first time you breathe in the influenza virus, in about 10 to 14 days, you make some antibody. If you ever are exposed to it again, you're going to have a much greater response. Same thing happens with the coronavirus. I mean, it doesn't matter what we're talking about. This is basically the same for any pathogen we're talking about. And then this would be the secondary exposure or any subsequent exposure to the same exact thing. And again, the antibodies are coming from our B cells, right? So the same holds true with the vaccine. We get the first dose, that's the primary exposure, right? A vaccine, the idea is we're giving a piece of the virus or some component that will give us immunity to the virus without actually giving us the full-blown virus that's going to make us sick. So our immune system is exposed here, and yes, it still takes a little bit of a lag period, about 7 days before you start to detect any antibody, and the peak is usually around 10 to 14 days later. And your antibody levels will go down eventually. This is normal, right? And then you get a booster dose. And for the two vaccines that are out, one is 21 days later, one is 28 days later. right? So you've definitely had your peak antibody production from the first dose, and then we give you, at this point, your second dose. And now that's your secondary exposure. Your immune system has been primed. It has that memory of the same pathogen. So you get really, really, really high antibody levels. And this is what gives you the protection, right? A lot of it has to do with these B cells. Where do the T cells come in? Back here. I'm going to erase a little bit of this. The T cells, some of your T cells are going to be able to kill off a virally infected cell. And that's really important because when we're infected by a virus, our cells become factories that churn out more and more and more virus, right? If that's happening in your lungs, every time you cough or sneeze or talk or sing or laugh, you're expelling virus that can be transmitted to others. So the B cells make the antibody. That's really great. The T cells are going to be able to kill off what's already infected. All right. And the last bit here I want to talk about is a little bit of the mRNA vaccines, right? So our efforts so far, this is a list that was updated as of 2 days ago. I don't believe anything has changed. And the only vaccines that are being used in the US are the Moderna and Pfizer vaccines, both of which are an mRNA vaccine. And that just kind of tells us what type of vaccine it is. And I'll explain that more in a second. There are other vaccines being produced. The ones here that let's see, I think all but this one isn't out yet, and this one isn't out yet. But all the others are out somewhere else and being used in other parts of the world, right? And there's lots of different ways to make a vaccine, but all vaccines essentially are doing the same thing. They're giving a little bit, they're training our immune system in a way that we get protection. We get those B cells, we get the antibody, we get the T cells that can kill a virally infected cell. We get all of that, but without getting the full virus that is going to give us a disease, right? So mRNA vaccines, what are these? This is our coronavirus, right? It's the full viral particle. This is the coronavirus. And this little squiggly line in here is its RNA, its genetic material. But the spike, right? These red spikes that are important for attachment to our cells, the spike is encoded for by a gene that is just a little piece of this genetic material, right? This whole genetic material, but just this red portion is going to make this red spike. So the mRNA vaccines take that, just that little piece that encodes for, it's the instruction manual for the red spikes, and we just clip that, and we're going to encase it in some lipid fatty layer, just like this yellow stuff over here. And this is what you're getting injected with. It's a piece of the genetic material that will be the instruction manual to make a spike. It's not the whole virus. It's not the whole genome. So it absolutely cannot give you COVID. All it is the instruction manual for a spike, right? And then what happens is, this is a great diagram from, I stole this from New York Times, but it's a very, I was gonna draw this. I was like, hey, they found it for me, great, right? So this viral particle, because it's made of a little lipid layer, it can fuse with our cells. So we don't need it to dock to any receptor. It's going to fuse into the cells where it's injected. It's going to deliver this little piece of instruction manual. And hey, RNA is something that our cells have anyway. So when we see it, Our cells are like, I know what to do with that. And they take the mRNA and we create the spike protein. This is the viral spike protein. And actually, it's 3 separate little subunits that all assemble to create a spike. So we make the spikes. Our cells are getting the instruction manual to make the spikes. And what do we do with the spikes? Well, we put them in the membrane, just like the instruction manual says. And now what happens? Now our cells are expressing a little component of a virus. And our immune system is so good at what it does, it is able to recognize that that is foreign, and it creates an immune response, right? Just like we would have had if the whole virus was there. We get innate immunity, we get adaptive immunity, we get B and T cells, we get their memory cells, we get antibody. And some of our T cells are able to kill this cell because it's saying, hey, you look virally infected, right? We get everything. And then we get the same, oh, that's really messy. We get the same antibody response, right? So with the first dose, it's going to take a little, and then we'll get an antibody response. And then with the second dose of the mRNA vaccine, we're going to get an increased antibody level right off the bat, shorter lag period, and this is what gives us protection. Right? You can think of it as if a certain level of antibody is needed in order to give you immunity or protection against the virus in the future. And the first dose maybe gets you up there where you should be, but again, we know that what is typical in a primary response is that it goes down very quickly. It usually requires that booster shot to get high levels that are sustained for a longer period of time. So, Pfizer vaccine, let me skip to this. Pfizer vaccine, let's see what we're looking at here, right? So on the y-axis, we have the cumulative incidence. This is the cumulative number of people that got COVID. We're looking at the placebo, which was just a saline shot in the arm, versus this BN162B2, that's the vaccine. So one group got the vaccine, one group got a saline solution. Neither group knew what they were getting, they just got the shot. And then we're looking at time along the x-axis here. So this is days after the first dose. So at day 0, they're giving that first dose. And then for the Pfizer vaccine, it was at day 21, they got the second dose. They're watching for COVID cases at this point when the paper was published. They were out to 120 days, which is about 4 months long. So they're looking for cases of COVID. That's really on this y-axis. And this is a little insert just showing the first 21 days because it's pretty crammed when it's all scrunched in the corner there. So they're just popping it into a blown up view here. So you can see that protection started, they said around day 7 is when they started to see some protection of the vaccine. And then out here at 105, 112, 119 days, there's quite a big difference, right, between the placebo group and the vaccine. And they said at 7 days after that second dose, out through the end, it was about a 95% efficacy. So that's really, really good for a vaccine. It is 95% effective at preventing COVID infection. Now, a couple limitations of this. Oh, this is pretty blurry here. But One thing that this data doesn't tell us at this point, one thing is we don't know the safety and efficacy. They said beyond 2 months after the second dose, I think it was. So they haven't studied longer than 4 months because they started their clinical trial, I don't know, in September or so, or October. So we're still waiting for the long-term data. Will the vaccine be effective in a year? We don't know that because it hasn't been a year yet, right? So because they had phase 1, phase 2, then phase 3 trials, that's why we don't have many months of data yet. But we have data on at least the first 2 to 4 months, and it does appear to be safe and effective. They also didn't look at children, pregnant women, and immunocompromised persons. So that's another limitation of the vaccine studies. We don't know whether the vaccine protects against asymptomatic infection because all they were measuring was symptomatic COVID-19. They weren't testing the participants every day or every week to see, do you have it or do you not have it? They were only testing if someone came down with symptoms. So we don't know whether it will protect against asymptomatic cases. If it didn't, then asymptomatic people could still transmit the virus. But we think it should protect against asymptomatic infection. And lastly, how to deal with those that miss a second vaccine dose. We know that you have to come in like at 21 days and at 28 days. If you can't do that, what does the efficacy change? Right, so Moderna is basically the same thing. Again, it's the cumulative event rate here. And we see at dose 1 and dose 2, and then over time, that the placebo group had an increased risk of getting COVID-19 because they had no protection, right? It was just saline. And the vaccine was effective. In this case, 93% or so effective. So both of these vaccines are very effective. Now, at this point, I think I want to stop. I've got some other things that I'm happy to move around, but I kind of want to see where the questions are at this point and let that decide where we go next. So I want to keep this up. So maybe if anyone has questions, send Justin a quick little notice and
Perfect. Thank you, Kara. Appreciate what a great presentation for a subject that all of us on this call this evening have been affected by in one way or the other, whether it's been in looking at the progression of a vaccine over the past 10 months or whether some of us on the call this evening have had a chance to get the vaccine. We've all been affected by this in one way or another. So I really appreciate the really great presentation.
Thanks.
So we do have a couple of questions. Katie asked, are there other similar vaccines that have a long-term effects that show up after 4 months of getting the vaccine? And right now, I know at kind of the tail end of your presentation, you were explaining how, we don't have a whole lot of data at this point, but, kind of that trajectory of what we've seen so far, are there other similar vaccines that we've seen?
Right. So that's a great question. And I know some people are a bit worried about, well, all we've been hearing all year is this is the fastest we've ever made a vaccine. And that can seem a bit scary to somebody that doesn't really know the behind the scenes. So let me first say that the Pfizer vaccine clinical 3 trials was with over 40,000 participants. The Moderna one was with 35 or 40,000 as well. So you won't be the first to get the vaccine, right? Even if you're a healthcare worker and you're getting it soon. And now we've had millions already get the vaccine. So in terms of the safety, In the past, when you look at vaccine history, there have been some very rare side effects that weren't detectable until such a huge percent of the population got vaccinated, because it was one in 10 million or something, right? So if you only have 40,000 in your trial and it's a 1 in 10 million side effect, you're not going to see it. So yes, there have been instances in history where there was a very rare side effect that required a big number to be vaccinated before we could see it. And that had been after several months. But I think with the number of people that are getting these vaccines, I mean, this is going up quickly in terms of the numbers getting the vaccine, that if there's a safety issue, we're going to see it. And so far, I had some data here on what are the side effects, what are the symptoms that people are feeling after they get the vaccine. This is Moderna's vaccine, and local events here just means at the site of injection. And they were looking for any adverse effect: pain, redness, swelling, or swollen lymph nodes in the area. And it was mild, moderate, or severe, right? So for Moderna, over 75% of people were feeling pain at the site of injection, compared to the saline was not very painful, right? Most of these cases were gray or blue, which means mild to moderate. Very little reddening at the site, very little swelling, very little lymph node, swollen lymph nodes. And then systemic effects that they were looking at fever, right? Very few people were getting a fever, and those that were, it was really coming at the second dose. Headache, right? Quite a few more. Fatigue, muscle pain, joint pain, nausea, vomiting, chills. You can see the things that they were expecting and they were collecting data on. And perhaps other people have heard that through the media, AstraZeneca paused their clinical 3 trials because they were investigating some neurological symptom. And then I believe they just came back on track when they determined that it was probably something that was pre-existing. So there's a lot of attention on the safety of these vaccines. And again, I think with how many people are getting it very quickly, we will see if there is any negative side effect besides these things.
Thanks, Kara. Janet has a question regarding what's the difference between the Pfizer, Moderna mRNA vaccine and Johnson & Johnson's vaccine.
Johnson & Johnson, are they on this list is coming up? Yeah, so Johnson & Johnson's, the AD26, this is a DNA vaccine. So there's lots of ways you can train the immune system in a vaccine there's lots of ways you can train the immune system with a vaccine. So the mRNA is one way, DNA is another, right? And it's basically doing the same thing. We're giving the code, the instruction booklet, to make the spike, so that our cells can make this viral spike, throw it up in our membranes, and attract all the white blood cells in our immune system that are saying, hey, that looks foreign, and let's mount an immune response. So it's a little bit different because it's DNA. I believe all of these are DNA vaccines, but the idea is the same. With DNA or mRNA, you are not, let me go back to this picture. With DNA or an RNA vaccine, it is impossible to be giving you COVID because, again, it is not the whole genome. It is just this one little part, couple pages ripped out of the instruction manual on how to make this thing. And because this one is RNA, it is not going to be able to get into the nucleus that has your DNA, right? Double-stranded DNA is in the nucleus. Because it's RNA, it cannot get in there, and it will not combine with our DNA. It will not be passed on when this cell replicates and makes one becomes two, right? So the RNA, you don't have to think of it as some like, oh, I'm going to have a viral gene as part of my genome forever and ever. RNA is very unstable, so it will be degraded very quickly. It's going to make a couple of spikes, put them up there, and then be destroyed. With the DNA, I believe, will be able to get into the nucleus, but it's not going to recombine and be part of your genome. So that the DNA is essentially the same thing. It's just DNA, and then it becomes RNA, and then it becomes protein. It's just adding one step. But you're not going to be some Franken monster. I know there's a lot of misinformation out there about how these work. Does that answer the question?
Thanks, Kara.
Yeah.
Another question that we have coming in is that, do you think children will be able to get the vaccination soon? I know that currently it's, there's some question that it's only for, adults aged 18 and older. What does that look like, over the next couple of months? What is kind of the processes of being able to at least offer a vaccination to, children?
Yeah, I'm not quite sure what they're planning to do, because we still don't know. The phase 3 clinical trials did not include children. It was 16 and older for Pfizer, at least, and I think that was the same for Moderna. So we don't have much data then on children. I do know, though, that school-aged children and even younger are not considered to be major spreaders. Typically, they have mild symptoms, typically. And it is a strong hypothesis at this point that mild cases are going to be able to spread the virus less effectively. If you have severe COVID, you are more likely to have higher viral loads and transmit and cause more severe disease in the people you're infecting, because they're getting a higher load. So it's something to think about. The more virus you breathe in, they think, not for sure yet, but they're thinking the more virus, the more severe. So mild cases are going to lead to probably more mild cases. Severe can lead to more severe. Depending on a whole bunch of other factors, right? How close you are you wearing a mask? Are you indoors or outdoors? A bunch of things. But children, again, typically are asymptomatic or only have mild symptoms. And if they're masked, then the risk of them transmitting is even lower, and they wouldn't be expelling much anyway. I don't know what the vaccine protocols for children will be. I don't know, because it's not FDA approved yet, right? We're still in emergency use authorization periods for both of these vaccines. But they do think they've been collecting data all school year, and they think that the opening of schools wasn't strongly related to outbreaks. So I think the first and foremost are the healthcare workers and the elderly are really important to get vaccinated. Yeah, sorry, I don't know more about the children's side of things.
Thanks, Kara. Another question from John is, is there cause for concern for a portion of the population that may decide, that they do not want to get the vaccination for whatever reason? Does that choice of not getting the vaccination compromise the the beneficial effects of individuals that have already been vaccinated?
Does it say that last part? Does it compromise?
So does it compromise the beneficial effects of the vaccine of people that have decided to leave it?
Yeah. So if you only get one vaccine, I think this Pfizer study. Let me erase some of this mess. The Pfizer study did say after one dose. Let's see, this was 82% effective. But they didn't look at this. There haven't been any studies where we just give one dose and we don't give a second dose, right? Because in the phase 1 and phase 2 trials, they tried different dosages. They tried different lengths of time. That's the more experimental point. Then they decide on a dose, they decide on a schedule, and now they give it to a bunch, thousands and thousands of people. So there hasn't been a clinical study with Pfizer or Moderna where they just give one dose and then pretend that nobody comes in for the second dose. So we don't have the data on that. But knowing what we know about the antibody response, we know that the first exposure, darn it, this happens in class all the time too. The first exposure is going to give us a nice response, but then die down quickly. And the booster dose, that secondary response, or the second shot that you get, is really going to bring your immune system up to a level that will give you lasting protection. So yeah, I think it's really important to give a second dose. I know there's some, I read a paper of like, should everybody be given a first dose before the second dose? And I don't think that's a wise idea either, because we know, this is what we studied. We know that 1 and then 2 at 21 or 28 days is 95% effective. We have no idea whether 1 is going, we don't know this curve entirely, right? Is 1 going to taper off and you'll have no antibodies by 2 months out? We don't know that, because everyone in those trials got 2 doses. So I would say, let's stick to what we know. And yes, if someone only gets 1 dose, they're not going to be as protected, which means if they're exposed to COVID, they might have a more mild disease, is what I would predict, because they have some immunity, but they might still be capable of having the disease, some symptoms that are unwanted, and transmission to others. So yeah, vaccine compliance will be important.
Another question, how long might it take for vaccination rates to have an impact on the infection rates, and kind of the trajectory that we're seeing, or that we've seen in the past?
The first part of that again, sorry?
How long might it take for the vaccination rates to have like an impact on the infection rates?
Yeah. At first, when they were studying when they were hoping for a good vaccine, they were saying, we'd be happy with like 70% effectiveness. And now we're at 95% effectiveness, which is great. We blew past the early hopefuls, and we're doing really great in that regard. But we don't yet know what percent of the population needs to be vaccinated in order to have herd immunity. I know everyone's heard of herd, H-E-A-R-D, of herd immunity, right? It's a real thing. It is the idea that if enough people in a population have protection, have immunity, it protects the few people that don't. And it is a real immunological, epidemiological principle. For different diseases, the percentage of the population that needs to be immune is different. So for example, for measles, which is another viral disease, it is actually considered one of the most infectious of the infectious diseases. And you need to have about 95% of the population immune in order to protect that 5% that isn't. So if you drop to 80% immunity in some communities where there's not high vaccination rates, then you have the potential to have an outbreak. So with coronavirus, SARS-CoV-2 virus, we don't yet know what percent of the population needs to be protected before we start to see this outbreak die down. So I could only guess. It could be something like 70%, it could be 80%, it could be somewhere in between. I don't know. But we do know in the meantime what small things can make a big difference. Wearing masks absolutely makes a difference. Keeping the 6 feet makes a difference, and avoiding large crowds, especially indoor crowds, makes a difference.
Thanks, Kara. Another one that just came in for, those individuals in the older population, have there been any research or any studies that have seen severe reactions to, the vaccines due to them having, an older immune system?
That's a great question. So Pfizer's vaccine actually they separated theirs out into 16 to 55-year-olds, dose 1 and dose 2, and greater than 55-year-olds, dose 1 and dose 2. So actually, this first slide looked at local events. So look to the right side here. This is 55-year-olds or older that were in the Pfizer vaccine phase 3 clinical trial. They do mild as green, moderate blue, severe as yellow, and grade 4. I don't know why they chose grade 4. That's really, really bad, apparently. So, and again, we're looking at pain, redness, swelling, typical. So yeah, in older people, there was 71% that got the vaccine. This is, again, just the name they were giving for their vaccine versus 9% for placebo. Geez, that's actually less than the 16 to 55-year-olds. And then, let's see, the second dose, it was 66% reported some sort of pain. When we look at the systemic effects, which, right, local was just kind of at the site. Now we're looking at things like fever, fatigue that are more whole body effects. For 55-year-olds, it looks like some fatigue and headaches after the first dose. Those were each a little bit increased after the second dose. And maybe some muscle pain and joint pain a little bit more in the second dose than the first dose. How do those numbers compare to the younger generations? They're a little bit different, right? These are even higher in the young people than here. So yeah, there's some differences between age groups. In terms of the immunity, though, I'm not sure that they separated it out to show how effective it was for each age group. Moderna did, though. Let me share my desktop a second. Moderna looked. No, this is Pfizer. Pfizer looked at vaccine efficacy based on age group. It looked like it was 100% effective for over 75, 94% effective for 65 and older. Males a little more effective than females. They did race, right? So they were really thorough. And that reminds me, I'll have Justin put into the chat some resources. I kind of pulled together some nice websites and resources for everyone if they want to learn more. And these papers that go over the phase 3 clinical data are in that resource document that I'll have him post. So another thing, I don't know if there are a bunch more questions.
We do have a few more, Kara, if we...
Yeah. I was gonna backtrack, but yes, let's address questions.
Perfect. Question from Peter actually is regarding some new investigation and success regarding moderators that I guess Merck is looking into. And I guess what it does is these moderators mitigate the inflammatory response that COVID patients have. Do you have any information or any knowledge regarding those at all?
Yeah, so there's a bunch of treatments that are in clinical trials. But so vaccines are preventative, right? Prophylaxis. Before you get COVID, they protect you. So once you have COVID, what can be done, right? So yeah, let's talk about some treatments. I don't have a blank slide here. Let me add a new slide. There we go. Great. Treatments. Okay, so the only FDA approved treatment so far is remdesivir. This was a repurposed drug, so it already had FDA approval for Ebola and Marburg viruses. And so it was already approved. They did the safety stuff, yes. They just had to do a quick clinical trial to make sure that it was okay to use and safe to use or effective to use for this new virus. So it went a little bit quicker because it was being repurposed for a new virus. A typical antiviral, and what it does is it changes the RNA replication. In simplistic terms, it interferes with making more of the same RNA piece to put in new viral particles. So this is the only one that is currently FDA approved, but there's tons that are in clinical trials. So yeah, when you are infected, we did mention that in severe cases, there's a huge immune response. And I can't spell and talk at the same time. And a lot of what you might be hearing about are these cytokine storms. This is at the point where it's no longer the virus causing damage, it's the immune response causing damage. So the cytokines, what is a cytokine? It's a little chemical signaling molecule. It's the words and conversations that one cell sends to another to understand what's going on everywhere. And a lot of these are going to be pro-inflammatory. One in particular that they know, and I'm going to be getting a little bit specific here. One of these pro-inflammatory cytokines that is elevated in severe cases is IL-6. It's just the name of this little chemical communicator, but it has a lot of effects that can lead to like sepsis-like symptoms, and which can cause multi-organ failure and circulatory collapse and whatnot. So one of the treatments blocks IL-6 from attaching to other cells, so they can't read the communication. There's other treatments that block the receptor. Oops. So some of them actually bind to IL-6, so it can't bind to a receptor. Here's our host cell. So for a host cell to respond to IL-6, it would have to have an IL-6 receptor. And when they bind, it says, and the message is relayed to that host cell. So there are some treatments. Whoa, what is going on? All right. Some treatments that block the receptor, some treatments that block the cytokine. There is, let's see, there's a bunch that block IL-6. I'm not sure which one someone is talking about specifically, but yeah, there are a lot of these small molecule inhibitors that are out there that either have specific effects on a specific cytokine, or corticosteroids are being used, such as dexamethasone, which in general calm down the immune response. So if we have this big pro-inflammatory response, some drugs are aimed at saying, just calm it back down, because it's the immune response going wacko that ends up causing a lot of the damage. So that's, I mean, I don't know specifics about Merck's drug or whatever, but there are a lot of monoclonal antibody treatments, small molecule inhibitors that are being either repurposed for treating COVID-19 or generated, right? So the convalescent serum, when somebody recovers from COVID, if you take the antibodies that were made in that person and put them into another, they should be able to do the same thing, is bind to the virus, and then there are mechanisms in the immune system to come through and eat it all up, degrade it, right? So instead of convalescent serum, some companies have said, well, let's just make the antibody that is going to bind to our RNA virus. So we can make antibodies. There are antibodies, 2 or 3 different types of antibodies preformed that are being explored in clinical trials. So in a severe case of COVID, these preformed antibodies may give a little boost that could help that person. So yeah, there's lots of different types of treatments that are being explored. So we don't have all our eggs in the vaccine basket. And along that line, we don't know what the best vaccine is going to be. The first 2 to cross the finish line here aren't necessarily the best. They were just perfectly primed to jump on this the minute we had the RNA sequence of this virus. They were the ones that were like, that's all we needed, here we go. And I think their clinical 1-phase trials for Moderna started like the middle of March, when most people were just catching news of this thing. So there are 43 different vaccines. This is from 2 days ago in phase 1, 20 in phase 2, 20 in phase 3, 8 for limited use, 2 approved, not in the US, right? We have, our 2 are in this emergency use authorization only. They're not FDA approved yet because the phase 3 trials aren't complete. These 2 are approved in other countries, and I don't know who the abandoned one is, but we've got a lot coming through, and they're all continuing through because they think mine might work better, might have long-lasting immunity, might be cheaper to produce, might be more effective to administer, right? Some of them are designing nasal sprays so that it works right in the place where it's going, where you are most susceptible to COVID, right, coming in, versus the intramuscular arm shots. So we don't know, we won't know for a long time which is the best strategy. But it's a really cool time in history because it's one of the only times when we have, how many, 90, I think at one point it was close to 90 different groups around the world, all trying to solve the same problem. It's going to be really cool to study which type of vaccine really is the best.
Thanks, Kara, for your explanation on that. We have 2 more questions. Question from Maggie: how different is this new mutation of COVID that we've seen recently, and do you think the current vaccine could potentially protect against it? If not fully, a small proportion. What are your thoughts to that?
Yeah, so the UK variant that everyone's been hearing about. We've just been hearing about it the last couple weeks, right? But really the UK noticed from September, like between September and November, they were seeing really quick rises in this variant. And so now we know that the variant is about 56% more infectious, more transmissible than circulating strains that we've got in the US. but they also know that it's not at this point leading to more hospitalizations. So what that means simply is it's not causing more severe disease, but it is more contagious, more infectious, which means that a smaller amount of it inhaled might be able to cause disease. Versus the strains now maybe have to take in a larger dose in order to get the infection. So there's the great potential that it could spread rapidly if it's more infectious and still lead to more hospitalizations only because more people are getting infected, right? So it's not that it's causing more severe disease necessarily, it's probably that it's spreading more quickly. So that is an important thing. In terms of the vaccine, they don't know yet. There haven't been any peer-reviewed studies. Early results, if you look for this online, as I was doing a couple days ago, they say early results are promising that the vaccines will protect against this variant. If it's an issue of transmissibility, what does that mean? It's a mutation in the spike. Actually, let me go back to this guy. Right, so it's a mutation in the spike that changes the spike conformation and probably means that it fits tighter, so that you can have fewer inhaled. If they fit tighter, they cause more disease, or they cause disease, period, right? But again, not a more severe disease, just likely that you don't need as much of it to cause disease. So if you're still spewing out the same amount, you're going to infect more people, right? So yeah, the early results so far say that the Moderna and Pfizer vaccines should be protective, but there's no peer-reviewed studies on that yet, so they can't say for sure. But again, we've got all these other types of vaccines coming. So even though the mRNA vaccines really are just about the spike, right? So if the spike changes a lot, we didn't have immunity to any other parts of the virus because we only had that spike coming in. Well, there's these other types inactivated, right? They're just taking the whole darn thing, inactivating it so it can't possibly infect your cells. And then you've got all the little foreign pieces in there. So I would say I'm not terribly worried because there's going to be a bunch of other vaccines coming out pretty soon. It's a good question.
And the last one from Kate, have there been any reports of any allergic reactions to the vaccine? And if so, do they know what caused those reactions?
That's a good question. So that's not something that was in this chart, right? Allergic reactions isn't on here, but it would certainly be something that would catch their attention or be required to be reported to the clinical study. So I don't know if there's been any, if there are, the people that are running that clinical study have been made aware of it. I would say if there was something that was very serious, they'd put the trial on pause until they could determine whether or not that response was due to the vaccine or whether it was due to some underlying condition or other exposure. But there's not a lot in this vaccine to cause an allergic reaction, right? The influenza vaccine is still made in chicken eggs, for example. So when you get the vaccine, they have a little disclaimer: are you allergic to eggs? Right? Don't get this one. We make it in another type as well that doesn't have that wouldn't have any egg proteins in the vaccine. I'm not saying there are egg proteins in it, but they purify out the virus from the chicken eggs, but it is made in a chicken egg. And yes, there's a potential that someone who's very allergic to chicken eggs could have a reaction. But with this mRNA vaccine, it's a little piece of nucleic acid. It shouldn't cause an allergy. and the little lipid coating, I'm not sure what that's made of, but I'm sure that it's something pretty inert. So yeah, I don't know anything more than that though.
Thank you, Kara, for answering, everyone's question here this evening, and for being so informative on like I said, on a subject that is so important to so many of us at this time. So before we end our night, I want to turn it over to my colleague and director of alumni engagement, Amanda Manza.
Thanks, Justin. I just wanted to say a big thank you to everyone for coming out tonight and joining us for this virtual Return and Learn. And a very big thank you to Dr. Mosovsky for putting all of this together and answering all of our questions. I think it was a fantastic presentation that gave us a lot of really good information. So kudos to you for all of that. And such a blessing to Moravian that we have such awesome professors that have such great knowledge and expertise in these areas. So definitely helpful during these times. I want to thank Justin Durenzis for making sure all of this works smoothly tonight. So he's the one that puts all of these together and makes all the tech work behind the scenes. So a big thanks to Justin as well. I just chatted out to everybody in the chat. Our next virtual Return and Learn is February 23rd on the social determinants of health with Dr. Sabrina Terizzi from our econ department. So I hope that everyone will register and join us for that one. If we don't see you before then, we look forward to seeing you that evening. Have a great rest of your night and stay healthy, everyone.